Hi, everyone. Welcome. Thank you for being here with us. Today I would like to invite you to join our line event about contributions of synthetic biology for human health. Thank you, everyone, for being here with us. My name is Caroline. I am part of iGEM Design League team, specifically working with partnerships. And I would like to say thanks to our partners, Ginkgo Bioworks, Synlogic, Instituto SENAI de Inovação em Biosintéticos e Fibras, and Instituto Tecnológico Vale. I would like to talk very quickly with you about iGEM Design League and iGEM Foundation, and then we start the event by inviting Jillian and Mona, our guests here. So iGEM Foundation is an organization, uh, is a nonprofit organization based in the US and Paris, that works for the advancement of synthetic biology education and organizes the largest global student competition on biotechnology. iGEM has been working for 20 years. This is the 20th anniversary, so we're really excited about it. And we have very big numbers and interesting numbers. Last year, we had more than 350 teams in the global competition from over than 40 countries. But it's a very big and non-biotech competition, but it's not global because, 40 countries is not everywhere. And recently, iGEM Foundation has a new purpose that is about to make sure that the field of synthetic biology and all of the power that this technology holds gets developed by everyone. Uh, this is how iGEM Leagues was born, and the iGEM Design League is a regional competition, part of this leagues program that focus on the development of synthetic biology in Latin America. This is a regional competition in which it's in the third year, and this year we have 25 teams in the competition from 10 countries, and our main goal here is to make sure that the field of synthetic biology grows and to kickstart the career of Latin American students on synthetic biology. And we provide tools and methodology to help them to be to like evolve their knowledge in this field. And wow, the competition is really great. We can send you the link of our website and our social media later in the chat. But we are here today to talk about synthetic biology for human health. The first question is, what is synthetic biology? We usually say that symbio, that is how we call synthetic biology, is a combination of biology and engineering to develop and to design biological system and its components. We use the knowledge of biology in a framework and a development cycle common in engineering projects. We try to standardize biological parts and try to combine them to make new biological system that doesn't exist in nature. All of that by looking for application and being helpful to the world. It has applications in many areas. Here we have a quick video from one of our partners, that is Instituto Tecnológico Vale. They have been working with studying the biodiversity of Amazon in order to learn more about the genetic richness and how we can use it as a source for synthetic biology projects. Chris, can you put the video on the screen, please?
Quando olhamos para uma floresta tropical como a Amazônia, é difícil colocar em números a grande quantidade de produtos e serviços que uma região como essa nos oferece. É como se estivéssemos dentro de um grande armazém. Aqui se pode ter serviços que vão desde a regulação climática até mesmo a produção de medicamentos. Hoje vamos falar de uma planta muito usada na produção de medicamentos e importante fonte de renda na região, o jaborandi. O jaborandi é um arbusto que cresce na parte baixa da floresta e é a única fonte natural de pilocarpina. A pilocarpina é o ingrediente tirado das suas folhas e que é usado na composição de vários medicamentos, como os que servem para o tratamento de glaucoma, onde é o principal medicamento, e também nos tratamentos de alguns tipos de câncer. O jaborandi verdadeiro ocorre somente em 3 estados brasileiros: o Pará, o Maranhão e o Piauí, e é uma importante fonte de renda para as comunidades locais. No entanto, o extrativismo desordenado e o avanço do desmatamento na região reduziram a ocorrência do jaborandi. Hoje ele é uma espécie ameaçada de extinção. Aqui no Instituto Tecnológico Vale, em Belém, juntamente com outros parceiros, temos desenvolvido pesquisas que contribuem para a conservação e o manejo sustentável dessa espécie. Nossos estudos começaram com o mapeamento das populações de jaborandi na Floresta Nacional de Carajás, um dos redutos atuais da espécie por ser uma área protegida. Nós também buscamos identificar plantas matrizes que produzem a maior quantidade de pilocarpina, o ingrediente dos medicamentos. Até o momento, temos identificado plantas com até o dobro dessa substância. É por meio dessas plantas matrizes que estamos direcionando a produção de mudas, tanto para reflorestamento quanto para plantio em sistemas agroflorestais na região. Para compreender como uma planta produz mais pilocarpina do que a outra, estamos buscando respostas no DNA da planta. O genoma completo do jaborandi está sendo montado pelo ITV em uma parceria com a Universidade de Copenhague, na Dinamarca. Juntamente com estes resultados, usamos dados de expressão gênica para identificar a rota de síntese de pilocarpina, ou seja, os mecanismos que a planta ativa para produzir este composto. A importância de descobrir essa rota é possibilitar o aumento da capacidade de síntese de pilocarpina, pois o jaborandi, quando cultivado, produz pouco dessa substância. Isso faz com que a gente precise continuar coletando jaborandi na floresta.
Well, this video tells us a little bit more about the work that Instituto Vale has been developing, and it's an example of how we can use our knowledge, our skills and tools of synthetic biology to develop the bioeconomy in Latin America. Well, today we're here with Julian Means from Synlogic and Mona Oliveira from Biolinker. They work in startups that are developing very nice tools and a very interesting work in synthetic biology. So I would like to ask Jillian to introduce herself and tell us a little bit more about Synlogic. Feel free, everyone, to send questions in the chat. And after Jillian and Mona's presentation, we will have a key questions and answer session. So they will be here to answer everything for us. So Jillian, thank you for accepting our invitation.
Thank you very much for having me. It's a very exciting chance to share more about Synlogic and the really exciting work that we're doing in the space of living therapeutics. So let's see if I can pull up my slides. While I do that, I will introduce myself. I'm Jillian Means, and I am a strain engineer at Synlogic. That means that I apply synthetic biology to engineering living bacterial therapeutics. So we engineer into an E. coli background, actually, to try to treat certain diseases. So that means moving around a lot of modular genetic elements between different strains to try to get to do what we want to see. So essentially transforming medicine through synthetic biology. Synlogic is really at the forefront of this application of synthetic biology to human health, particularly in bacterial therapeutics. So our mission here at Synlogic is to treat diseases, particularly those that are underserved by other modalities, by researching, developing, and commercializing a new class of medicines through this application of synthetic biology. And how exactly does that make us stand out? So we call our living therapeutics synthetic biotics. That's just our own personal term for it. And we develop them by applying precise engineering to address specific disease biology in this bacterial strain. So this is a little bit different than other bacterial therapeutics that are on the market or in development There are quite a few that involve things like full microbiome transplants, maybe for the treatment of C. diff. There are consortia of very specifically chosen microbiome members, and then there's also single strain probiotics. These are all complex and they're microbiome focused, and they do tend to have more broad biological effects because they're relying on strains that just exist as they are in nature. At Synlogic, we approach it from a different angle. We say, okay, here's this probiotic. How can we make it do this extra function that we want it to do in order to really attack this validated biology that's underpinning the origin of these diseases? So we do this by applying the engineering principles of synthetic biology, and that allows us to be microbiome independent. So what that looks like in more specific terms is we have this product engine, which is a non-pathogenic probiotic bacterial chassis, which is E. coli Nissl. And we use this programmable precision engineering to create this new, safe, orally administered and locally acting candidate to target these unmet diseases. And we actually have clinical validation in a few particular diseases, specifically PKU or phenylketonuria, enteric hyperoxaluria, and homocystinuria. These are all fairly rare or very rare in certain cases metabolic disorders. And I'll go into PKU and homocystinuria or HCU in more detail later. But we're able to use this to advance this new modality of transforming toxic metabolites or producing active metabolites that this E. coli natively would not be able to do. And by using this, we can actually impact diseases in a meaningful way, or we hope we can. So to get into a little bit more of the engineering for specifically what we can do with our platform, I already mentioned metabolite consumption. So that is actually our core function. For these metabolic disorders, they tend to be characterized by an overabundance of a particular metabolite. And so we've been able to engineer our strains to actually degrade these metabolites into non-toxic byproducts that then can hopefully ameliorate the effects of these diseases. But we've also shown that we can produce small molecules, maybe receptor agonists to incite a certain host function along that line, as well as display different proteins on the surface to decorate the cell for certain functions, like maybe binding, as well as secreting peptides and other protein effectors, once again, potentially for receptor binding to elicit a host response. So these synthetic biotics are non-colonizing, so they are transient, and they don't rely on making a niche in the microbiome, which makes them actually more versatile. And it also allows them to have predictable drug-like properties. They have very limited risk of genetic drift, and we use reusable genetic parts, which allows us to build a very robust pipeline through learning and applying of these parts and other things like manufacturing across a lot of different programs. So what does that pipeline look like? I'll go through it right here. We have, as I said earlier, mostly focusing in on metabolic disorders. So phenylketonuria, PKU, homocystinuria, HCU, both of these are characterized by an overabundance of a particular amino acid. And therefore, the strains have been engineered to consume this amino acid to lessen the burden on the patients. And I'll go into more detail on that later. Our current lead candidate in our lead program is SYNB 1934. It was actually awarded a generic name recently, which is a bit of a mouthful, but I'll do my best to pronounce it. It's lavafenagine marcelicobac. I'm probably going to just use SYNB 1934 going forward, because that is quite the mouthful. That's currently entering phase 3 trials. It actually, the phase 3 trial started about a month and a half ago, ramped up. So that is very exciting to see that we're finally getting close to validating all the way that hopefully this will be successful and that we'll really be able to impact these underserved patients. SYNB 1353 is in some ways the successor to PKU. Homocystinuria is also characterized by an overabundance of amino acids, and it has finished up phase 1 and will hopefully be going to phase 2 in the future. And then we also have a couple other programs that have either completed phase 1 or are in IND enabling studies to get to phase 1, and a few other preclinical pipeline programs, cystinuria, and a collaboration with Roche on inflammatory bowel disease. So now we'll get into some details on our lead program with SYNB 1934, which has the potential to be the first and only oral non-systemic biotherapeutic available for this treatment of PKU. So what exactly is PKU? PKU is a disease that's an inborn error of metabolism. So essentially these patients have a defect in their PAH enzyme, which is responsible for metabolizing the amino acid phenylalanine, or Phe, that's found in dietary protein. Some patients have completely non-functional enzymes, and some patients have some functionality, but it's still not enough, not up to the level of a healthy individual. And when Phe builds up, it actually becomes neurotoxic. It leads to intellectual disabilities, deficiencies in mental processing, social engagement, emotional problems. And these neurocognitive symptoms are not just the beginning of the burden, too, on these patients, because in order to manage this disease, the only real treatment, there are adjunctive therapies to try to rescue the PAH enzyme activity if there is some residual activity, but the main treatment is to simply restrict protein in the diet. And so these patients are unable to eat more than 4 to 6 grams a day of protein, which is an incredibly low amount. They have to get prescription low-Phe foods and track religiously what they eat day in, day out, as well as take supplemental amino acid medical formula that adds back into their diet the non-harmful amino acids that they need. This diet is extremely challenging, and approximately 80% of patients have Phe levels that are higher than their goal. And once the Phe levels get higher than that, they start to see these neurocognitive symptoms. So SYNB 1934 is designed to consume Phe to try to meet these patients' needs. It targets both dietary and circulating Phe in the GI tract. So both Phe that's originating directly from protein consumed in the diet, as well as Phe that has actually entero-recirculated, as we call it, from the bloodstream back into the small intestine. So that's able to access a fairly large reservoir of Phe from the body, just from the GI tract. Our E. coli Nissl chassis has been engineered for SYNB 1353 to have 2 Phe-degrading enzymes called PAL and BLAD, as well as an importer to bring more Phe into the cell so that these enzymes can have more access. And these produce non-toxic byproducts in the gut, which are then harmlessly excreted by the patient. It's oral administration, and it's non-systemic. It stays in the GI tract, which is a very good advantage for these patients. So this is the phase 2 data for SYNB 1934, and it has demonstrated potential Phe reduction within PKU patients here. We had a 60% response rate, so 3 out of 5 patients dosed with SYNB 1934 achieved at least 20% Phe lowering compared to baseline. And of these 3 patients, they had an average lowering of 53% Phe, which is very good, as well as one patient in particular that had actually 80% Phe lowering. The graph in the middle you can see is actually singular patient data separated out. And that 80% lowering was actually in a patient that was already on Kuvan. As I mentioned earlier, there are a few therapies that can try to work with existing PAH activity, and Kuvan is one of those. And now for a little bit of detail also on HCU with SYNB 1353. So HCU is in some ways similar to PKU. It involves a amino acid that is toxic and builds up to high levels due to an inborn error of metabolism, where a native enzyme in a human is there's a mutation that causes it to lose function either entirely or in part. In this case, it's actually not quite as straightforward, because what happens is you bring methionine in through your diet, and that is an essential amino acid, which is then converted to homocysteine. And homocysteine is actually the bad actor in this disease. The cystathionine beta synthase enzyme is inactive, which prevents homocysteine from being converted further into from this cycle, and it builds up in the body. When you have too much homocysteine, it leads to developmental delays, lens dislocation, skeletal abnormalities, and most frighteningly, a significantly increased risk of stroke, sometimes at very young ages. So this is a very serious disease. And similar to PKU, we've engineered a bacteria to try to meet the need of these patients. Just like PKU, they have to restrict their protein intake. By lowering the methionine that is taken in, it lowers subsequently the amount of homocysteine that is circulating, and therefore that is how they control their disease. They have to take the supplemental formula. They can only have a few grams of natural protein a day, and it's just a huge struggle for these patients. So we have 1353, which has an enzyme, METDC, that degrades methionine into a harmless byproduct that cannot be transformed downstream into homocysteine, as well as an importer to bring more methionine into the cell. And this was actually developed in partnership with Ginkgo Bioworks, which is really exciting to show how we can work together with synthetic biology to get this really great potential treatment to patients. And we've actually found that in healthy volunteers in a phase 1 study, we can see some methionine lowering after a methionine challenge in the plasma. These healthy volunteers, they're healthy. They have an active enzyme. And so to be able to see any lowering at all is really exciting, because you're sort of fighting against the body's natural, really good and efficient ability to really normalize these amino acid levels. So this is really exciting, and we're hoping to move to a phase 2 proof of concept study in patients in the future. And really briefly, in the interest of time, I'll touch on some of our other pipeline programs that I mentioned earlier. These are all in the metabolic space. We have enteric hyperoxaluria, where there's an excess of oxalate. And so we have a bug that's been engineered to consume oxalate, and this prevents kidney stone, or would hopefully prevent kidney stone formation to help with chronic kidney disease. There's gout, which is caused by an overabundance of uric acid in the plasma, and by hopefully having this bug that can decrease the uric acid by consuming it in the gut, and by lowering it, therefore, in the plasma, it can prevent this buildup. And when you have too much uric acid, it can actually crystallize in your joints. And as you might imagine, crystal in your joints would be very painful, and it can lead to very debilitating arthritis. So that would be extremely exciting. This is in IND enabling, and will hopefully be going to phase 1 at some point. And we also have a program in cystinuria, which is a more early stage. It's also targeting methionine. So to wrap it up, Synlogic is a clinical phase company. We're in phase 3 with our PKU program. And we have a robust clinical pipeline in the rare metabolic disease space that reflects our validated synthetic biology product platform of oral, non-systemic, novel biotherapeutics. And thank you very much.
Thank you, Jill, for the great presentation and get to know more about your work in Synlogic. I would like to ask everyone to let questions in the comments. Feel free to ask, I don't know, about technical stuff or maybe anything else. Feel free to ask questions. And I would like to invite Mona to join us here and tell a little bit more about Biolinker.
Hello, hi everybody. So I would like to thank you, iGen Design, to invite me to do this presentation. It's always an honor to present Biolinker in the CBU community. Okay, I hope everybody can hear me well. Let me share my presentation. Just a second. Right. Okay. Can you see my screen? Oh, nobody has me. So
yes, we can see it and hear you.
That's perfect. So today I'm here to talk about cell-free protein expression. But to talk about that, how we are working here in Brazil and Latin America, actually, to use this tool to create solutions for human health, for agriculture, for cosmetic, for many another fields. But I will focus today to talk about cell-free protein technology, how the BioLink evolved that here. And then I'm showing some examples and also how we contribute to healthy human health with our solutions. Okay. And to talk about cell-free protein expression, I'm starting with this short in the abstract resume. Everyone knows it's more tradition to do protein expression in cell-based systems. You do have many steps. We need to do transformation, to grow the cells, to express and induce protein production, lysis, centrifuge. And when you do downstream process, we can have many others protocols to purify your protein. So many steps, many different protocols, and this is the combination to fail. That's why express protein and establish new bioprocess and engineering process has a huge failed and is still in 50% up to now. And when you try to scale up that, the failing process also is increased. When we talk about cell-free protein expression, we invert a little bit this. Our company, for example, has an E. coli cell lines genetically modified that overexpress some enzymes responsible for protein expression systems, or like polymerases or chaperones. And we do have this cell line here. But you can do with E. coli, a normal E. coli, for example, and you grow these cells before lysin and extract these enzymes. These enzymes, this lysin, it's now in the tube, and you can now combine with energy subtract amino acids, salts, and it's a formulation with more than 16 ingredients. And do very fast your protein with this combination. You do like a micro reaction, like enzymatic reactions in vitro system. And using this, you can express, just put your DNA or your template on the plates and express in hours. And it's so practical and easy to use that this kit can be applicable for education or to toxic proteins that's hard to express in cell-based, and also difficult to express proteins since we don't have a more live cell here. and we eliminated some points. Also, with this technology, we can study deeply how elements is important to protein expression, how to regulate the ribosomes, how to improve the polymerase efficiency and polymerizations process, promoters. It's so nice and advanced that I'm in love. I love this technology. That's why when I test that during my PT, I said, why nobody using that? Why people don't know? Because Biolinker didn't invent that. It was the first time he used it to determine the genetic code for Nirenberg. He used E. coli extracted, and he did, at that time, the first cell-free protein expression system. But it was so hard to express at that time. This is not become a product, was used only for research process and development. And during the time, it was improved. The process improved, the formulation improved many things to create what we call now cell-free protein expression systems and kits. And here at Biolinker, we made one own formulation, and we do have these kits to do protein a fast and easy way, using different ingredients and using this modified E. coli. That's special. Our kit was launched in 2019. The company started in Latin America. We got our first investment to create the kits here. And we starting to study how to lyophilize these kits and increase the stability, increase the expression amount, and use these kits to accelerate the process to obtain your proteins. That's the principal aims to use this. Also, we develop a new vectors. It's Biolinker. It's a vector that improves the expressions, has some promoters that can be used together with our kits to express any proteins using the platform. In about 2020, Natalia Cummins, it's another researcher in our team. She is very specialized to do lyophilization process, and she's starting to working to create the first version of our lyophilizated cell-free protein kits. Why? Why this is so important? It's so hard to do this kind of lyophilization. Also, this kit needs to be in very low temperatures to transport, to have a good performance. And when we talk with the customers, a lot of customers say that don't like the technology because they pay a lot to have the kits. And the kits, when coming to Brazil, because all companies that do or made these kits is outside from Brazil. It was too probably we have a lot of problems to import that the kits lost the activity. And then we starting to work in to make improvement to lyophilization, also to improve the logistic process, to our customers have the best experience using the technology. And the second reason to do that project was and we got it to do lyophilization cell-free protein kits. But the second reason was to use into education. And we started to do a cooperation with USP, the team from ECB. It's the Health Biology Institute here. They have a project called Biosynthesis Mirroring it's in Brazilian, it's bioscientist. I don't know how to translate that, but it's for children's on the school, on the middle school, underground. And they can use the kit without trying to eat. It's very important to be lyophilizated. Also, to go to schools that don't have labs or minus 18 ultra freezes. That's important to keep these kits. And that's why we're working to improve stability and create the best cell-free protein expressions kits to be used abroad in Brazil, mainly because we are continental country. And this kit now will be the first in this year will be used in the first school. And we are very, very excited to see how it's going to work here with the children's. But we already used the kit with underground and graduate students from University of ABC here in São Paulo. Also used that with SENAI and Biotechnology Institute here in Brazil. And Biolinker don't working only with cell-free protein expression kits. We start with that, but when you talk to the customers outside, we discover that people need the proteins. They don't know how to do proteins. They don't have all equipments to do proteins or test. And also it's expensive to do proteins when you don't know how to do. You need so specialized people to work on that. That's why Biolinker so watching so opportunity to work in to deliver protein with their customers. And we start to do on demand. And then come the COVID-19 pandemic situation. And we start to do COVID-19 protein expressions. And this was great situation for the world. And mainly for Biolinker also, we got a lot of projects. We start to make in 2 months, we have 9 proteins of COVID-19 to delivery for many universities, and some professor working to make a new test, a national test in Brazil to, they call popular tests for COVID-19. And also we work here with hospital to deliver proteins, to make ELISA tests. And my first time I went to television to talk a lot. I scientist, I was never been there, and it was amazing to show that we can use synthetic biology to help in the situation, but to create solution fast and easily. And to show that, for example, in COVID-19, as I told, we express 9 proteins. One of them was very hard to express. And then we have tried many cell lines. We first start with our cell lines. That's more cheaper. We want to do a cheaper process to create very low-cost proteins to deliver our customers. And then it didn't work well. And then we tried to do with cell-free protein expressions. And then you got it together, the monomerous proteins without cleavages. The proteins has a kind of problems that even we changed and use many protocols. They degrade during the process. And even to us that very specialized teams have a failed process to establish a new bioprocess. And that case I told you was difficult to express proteins, but sometimes it's hard to purify. It's difficult to create a way to have a good stability for your proteins. So proteins also degraded or need special buffers to have your activity or create the best environment to work on. And the principal point of the fields is the customer that don't know how to do proteins. There are few groups on the world working online to express proteins and improve bioprocess. And here at Biolinker, we pivot. As I told, we start with cell-free online, looking to sell online kits, but then we become a company that we discover new proteins and develop and design since in the DNA the new proteins and manufacture proteins for big companies, small companies, in university here in Latin America. And that's why we have got a lot of awards. Here we are award France in 2019. Now we are top 7 best biotech in Latin America. Also, we got in Poland over Summit as best startup in biotech. And many others I'm gonna show you fast. And we, in these 4 years of operation, we are proud to say that we are working with more than 100 proteins. And from this many different kind of proteins, has proteins for plants. That's very interesting, proteins that we express using E. coli, from fungus and parasites, from animals, from like pig, chickens, many others, and some ones for humans proteins, virus, and bacteria, and even proteins that don't exist, or completely synthetic. We have done some ones to fiocruz here in Brazil, for example. From this more than 100 proteins, there are some proteins that never been expressed before in E. coli, so we are the first one to express. Also, proteins from the biodiversity in Brazil. For example, one that I really love it's from the most bright fungi in the world. It's from Brazil. It's from Piauí, and we are expressing that in your lab, the luciferase from the fungi. And right now, the most valuable is our platform. We evolved to create this platform that we have this genetic repository with many promoters and plasmids and vectors that we can be used to express proteins. The both platform to expression, cell-free and cell-based. And we are always doing the screening when we have when we are choose to do the protein for our customers. We design the way to get at the most increased amount of protein and the most purity protein in this process. Of course, there are some customers that don't need high purity. It need increase of amount. So we working as customer modularity process to create a tailor-made solution for our customer. Also, we have a different methodology of purification if the purification is the problem. And the last one, that's very nice. We have a cell line that can do mutations, aleatory and randomic mutations and on plasmids, on DNA. And then we can create in different clones and do direct evolution to create mutation of the proteins, mutate proteins that increase activity, stability on pH, stability in different temperatures to create the best ingredient to use in industrial purpose. And as I told you, we got a lot of global recognitions. On this year, we are finalists on Fellingwell Labs, and we are also it's going this process. Fellingwell, it's in Germany, and it's semi-finalist, and Haiku is from China. We have grants and accelerations and partners with some big companies here in Brazil, like Ambev. We are doing proteins to use in the bee, and we already have accelerated to create new ingredients for others food companies, but it's not the focus right now. This is our team. 8% of our teams are scientists from Brazil, almost PhD in biotechnology and biochemistry, working with the team. It's an amazing team to transform and increase the use of synthetic biology in Latin America. And we also have an investor, Fabrício Blois. He is a founder of iFoods, is a unicorn here in Brazil. And right now, more than 17 clients was impacted with our solution. And we export last year protein to Italy, and we do have some international customers like Roger, Merck, that it's partner with us to use the proteins using your platform. And this market is really huge. I think.
Oh, no. Sorry to interrupt you.
It's the last one slide.
Okay, yeah. I was to ask you if it's gonna take many minutes, but feel free to finish. Don't worry.
Okay, so it's the last one. I would like just to say that's a huge market to many other companies also coming, because we don't know a lot about different proteins from the biodiversity. For example, I like a lot this work that's showing that we have only sequencing 0.2% of all genomes of eukaryotes in the world. So we need to know, we have a lot of things to discover, to develop, and the impact of bioengineering, it's calculating trillions for the next years. So thank you so much. This is my contact. I would like to invite all to visit us, and if you need to talk to me, I am available in many social media.
Yeah, it's a good practice or a good idea to follow Biolinker and also Synlogic in social media. So, Mona, thank you so much for your presentation. We have one question here in the comment box from Abby. Let's see what he asked. This is more for Synlogic. So, Abby says that his team is working on synthetic probiotic as complementary treatment against Alzheimer's. Wait, sorry. To prevent dysbiosis, we are considering a synchronized lysis systems using quorum sensing. Should we stick to that or switch to a non-colonizing bacteria strain? What do you recommend? Yeah, Jill, I think it's on you.
Yeah, that's a good question. It's a big question, and it's difficult to get at a solid answer without, I think, having much greater understanding of your platform, what exactly you're engineering your probiotic to do. We in particular at Synlogic, there are several reasons that we've stuck to non-colonizing bacteria. One of which is that it's just a characteristic of the particular probiotic that we chose. E. coli Nissl used to be, back in pre-industrial times, a native member of the human microbiome. And because it's E. coli, that gives us access to the much larger suite of engineering tools. It's not quite as easy to work with as lab-based, domesticated strains of E. coli, but it's still considerably more accessible than some other microbiome members. However, with the modern diet and modern humans, it doesn't colonize us anymore. There's a lot of speculation as to why that might be, but because we chose this chassis, that was sort of a flow-on effect, but it also wasn't necessarily bad for us. Because we're trying to treat these metabolic disorders, we need to be able to understand how our bacteria is acting in the gut. And by having it not colonized, that makes the pharmacodynamics much, I wouldn't say simpler because it's still complex, but easier to track and model, because we know how much is there, we know approximately how long it stays there, and we know approximately how active. So that's why we decided to go with this. A synchronized lysis system using quorum sensing could definitely be really interesting to keep your bacteria in check, to make sure that you're not throwing off the microbiome entirely. Another challenge to look into, though, would be regulatory realities of your particular space, but definitely could be the greatest challenge.
Yeah, following the question, what are the greatest challenges?
Honestly, some of the greatest challenges have been the strain engineering and the synthetic biology. Building, I won't say that's the easiest part, but it's more straightforward. You have the parts that you want to put together into a final product, and then we can do that, or we can't. Sometimes it doesn't work. But the regulatory aspects are often challenging because it is such a new field. That's not my wheelhouse, so it's hard for me to speak too much to specifics. But because there aren't super strong guidelines in place already, so it's sort of a learning process, which can be actually a benefit, but it is something to keep in mind. And also manufacturing the bug so that it can be taken as a drug. Going from a vial in a freezer to something that you can actually give a person is a really big step, like a really big. I don't think Synlogic knew how big of a step that was when we started. So definitely something to.
Okay, yeah, thank you. I have a question here that works for both of you. Mafer is asking about internships, or as a chemist student, or if she could take other courses, or how can how to be prepared to work on synthetic biology. So Mona, if you wanna give some tips.
Yeah, we have an internship here in Biolinker. We right now we're gonna receive one student from France. He's coming in August. I think the way to write, it's important to go to website and writing to us from your friends @biolinker.tech and send an email with curriculum so we can come back to you. We have a lot of projects, so it's really interesting. We like more students that can come in to spend more time with us, but we know that sometimes they cannot, because sometimes you need to have to real learning, you need to be more time. But usually they come in and spend 1 month with us.
Great. And Jillian, do you have any tip for internships or any opportunity? People are asking about it.
Yeah, unfortunately, Synlogic is not currently accepting internships. With the phase 3 trial going on, it's more focused in the clinical direction at the moment. However, definitely follow us and keep an eye out in the future, and feel free to reach out. And if something ever does open up, that we'd definitely be super excited. Could happen in the future.
Yeah, so everyone, you should like pay attention to the opportunities on the website and maybe write them with your curriculum. You can find an opportunity. Mona, I would like to ask you about your experience. About you have been doing many things. Like you started with Selfree, and then I know you for some time. And we are both Brazilians, and I saw like you launching new products, new services. And what are you doing now? You showed a lot of things here, but what do you do more? Like self-free expression or kits for, or maybe the platform? How is your pipeline working? And how is
We
and this is really interesting that you look to the what the client's looking for. So how is your process for the developing these new processes?
Very interesting question. Like we're still working with cell-free protein expression as innovation product because we believe in the technology, and we do know that there are many points to improve. And nobody have it done in the world, so we are working and following that, creating new formulation, new ways, new tests. But this is intern. Like we are working today to change it tomorrow. And as and company need to survive, so we have segmented like commercial part to working with the customer right now. What we can do right now to solving the problem of that. So 90% of our revenue comes from the project for research and development of new proteins using cell-based mailing, but sometimes high cell-free too. So it can be tools for our customers. Depend, of course, how many they have, how fast they need to be. Cell-free is more fast, but more expensive than cell-based, so it can be an option.
Looks great. Yeah. I will ask you the last question. We don't have any other question here in the comment box. I would like to know uh, your thoughts about the future of synthetic biology applied to human health. Biotech is evolving, synthetic biology as well is evolving so fast. What do you see for the future? What do you believe is going to happen the next few years?
Who's gonna fit first, you or me?
This is a hard question, I know. But since you're experts, I am really interested to know your opinion.
I can try to go first if that works.
Yeah.
Synthetic biology in the next few years, I think we're definitely going to see a lot of improvement. I think it's hard to say, in my opinion, whether or not we'll actually be more applied yet, but I think there's going to be a lot more in the works, in the pipeline. A lot of different new ways of looking at it. Like Synlogic is just looking at bacteria and engineering bacteria to do things in a specific context, but I think that concept can be applied, and there are startups out there that are working on similar things. So I think that we're going to see a really big broadening of the pipeline in the next few years, and who knows, maybe a few things will succeed, make it all the way, and that would be hugely exciting. But I can say that in the Cambridge area, in particular, in Boston, Massachusetts, Cambridge area, where I'm at, we're very close to MIT and Harvard, like they're right down the street. And I can say that both of those, they spin out a lot of startups, and they have also both recently, particularly MIT, started new synthetic biology departments, which I think is really telling that it's starting to come up from, starting in academia and then kind of move over. Either there's going to be a lot of really cool stuff in academia, but also moving over into industry.
Thank you, Jill.
So I am really enthusiastic about the future of synthetic biology. It's like we have a lot of things to discover and develop. And here, using AI, for example, to improve that, to improve to design better proteins in my field, for example, or to create solution to help the customer how to design or how to discover the best way to have proteins, for example, and looking for data about the production and predictable production and purifications process that we don't need to do many tests on the lab and not spend a lot of money try and get a fast results. So I really believe with this new tools from AI using to biological proposal will be the future and will be increased and accelerated the solutions that from academy to real change the society and create new products and create new solutions. You know, and here in Biolinker, for example, we're gonna work here with AI in the next month and using our database into protein expression and improve that to find some partners and some solutions to improve the way to express protein, for example. This is the one. We have many other fields to go, and and I think we need to change the way to create things biological. For example, we always working in cascade is different. For example, when we work into in information and technology, bro, they do ways more fast because they can have a separated process. And in biological, no, we do in cascade. We need to finish one to start another. But when we know all deeply about this, we can maybe separate that puzzle and working more fast, like we did, for example, to the vaccine for COVID-19. So we have a lot of things to improve to create real solution to go to the market more fast.
Nice. Look, how cool you're gonna work with AI. AI is in
Yeah,
everything today, so yeah, we can be
yeah,
behind.
exactly.
Yeah. So before we finish, we have just the last question here. It's for you, Mona. Wait, sorry. For Biolicare, if you happen to know the cost part of setting up these biotech platforms to produce your services, did you as a company have to import a lot from abroad? This is a question, important question
That's
for
really
us.
great question. No, actually, I import only for cell-free protein expression some purified ingredients that I need to do my formulation don't have production here in Latin America, so I need to bring from outside. That's why cell-free is more expensive. But for cell-based, no. Actually, we are one country that produce a lot of saccharose and mediums, and we have a potential here to go and lower the cost a lot, for example. And I have a challenge here to set up a lab, equipment's coming from outside. Yeah. And the capex of the company for the capex to do a lab here, it's very complicated, but the government is helping. It's helping. That's why BioLink is exist. Yeah, we're working. We have a lab, certificate lab, with clean room, all building with government money, investor money, and also with our customers sources that pay us to do the services.
This is really great. Thank you, Mona. Thank you, Jill, for being here with us today. It was really nice to hear from you, from expert, from industry. And thank you everyone who is still watching. We will send you later an email about feedback satisfaction survey, so please answer. This is important for us. And I would like to invite you all to get to know more about iGEM Design League. And also, if you're a company and wants to partner with us to keep empowering Latin American students to design with biology, feel free to scan here or to reach me in email or any other channel you want. So thank you
Oh,
everyone for being here. And
Carol, before
okay,
you
okay,
finish, just I saw a question about Gustavo. I would like
okay.
to say that anyone that want to do cell-free protein expression project in iGen can contact us. We can give the kits.
Oh,
So,
this is a good news. So,
yeah,
Gustavo,
would love.
write Mona. You might have a gift for your team. So,
Yes.
yeah, thank you, everyone. Mona, do you, if you want to say goodbye, this is the moment.
Yeah, thank you, everyone. It's been really awesome to be able to be here, and it's been great.
Okay.
Thank you so much, guys. It's really great. It was nice to meet you too, Gillian, and see your
Yeah,
work. It's really
definitely.
nice.
Meet you too. It's been
Yeah.
really interesting.
Bye.
Honor.
So, bye. Thank you.
Bye.